Hi everyone, welcome back. In this video presentation, we are going to discuss one of the most important molecules of adaptive immunity, that is antibodies. Antibodies are the major weapons of humoral immunity. They are produced by activated B lymphocytes, known as plasma cells, and their main function is to recognize and bind foreign molecules called antigens. Once antibodies recognize and bind to the target antigens, they can neutralize pathogens, activate complement proteins, and help other immune cells such as macrophages remove invading microorganisms. The outlines of this talk are 1. Basic structure of antibodies, 2. How antibodies recognize antigens, 3. Functional regions of antibody, and 4. Antibody isotypes. This is Dr. Mo. I'm Associate Professor from Newcastle University. First, let's look at the basic structure of antibodies. An antibody molecule has a characteristic Y-shaped structure. Each antibody consists of 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains. The 2 heavy chains form the inner part of the Y-shaped structure, while 2 light chains are located on the outer arms of the antibody. These chains are held together by disulfide bonds, which maintain the overall structure of the antibody molecule. There are 2 types of light chains, kappa light chains and lambda light chains, based on the amino acid sequence. An individual antibody molecule contains either 2 kappa chains or 2 lambda chains, but not one of each. Now, let's examine the structure of each chain in more detail. Both heavy chains and light chains contain 2 important regions, the variable region and the constant region. The variable region is located at the tip of the antibody arms. In the diagram on your right, you can see variable regions in green color. It is called variable because the amino acid sequence differs significantly among different antibodies. This variation is extremely important because the variable region determines which specific antigen the antibody can recognize. Different antibodies have different variable regions, allowing our immune system to recognize millions of different antigens. In contrast, the constant region has a relatively stable amino acid sequence. In the diagram on your right, you can see constant regions in yellow color. The constant region does not determine antigen specificity. Instead, it determines the biological function of the antibody, such as interaction with immune cells or activation of other immune mechanisms. Now, we need to understand 4 important terms: VL, CL, VH, and CH. The light chain contains 2 regions. The first one is called VL, which means variable region of the light chain. The second one is called CL, which means constant region of the light chain. Similarly, the heavy chain contains VH, which means variable region of the heavy chain. It also contains CH domains, which means constant regions of heavy chain. Depending on the antibody class, a heavy chain usually contains 3 to 4 constant domains. The combination of the variable region of the heavy chain and the variable region of the light chain forms a very important structure, the antigen-binding site, which is shown in red color in the diagram. Now, let's discuss how antibodies recognize antigens. First, we need to understand the difference between an antigen and an epitope. An antigen is any molecule that can be specifically recognized by an antibody or by receptors of immune cells. For example, a protein on the surface of bacteria or viruses can act as an antigen. However, an antibody does not recognize the entire antigen molecule. Instead, it recognizes a specific small region of the antigen. This specific region is called an epitope or antigenic determinant. Therefore, we can imagine an antigen as a whole object and an epitope as a specific part of the object recognized by an antibody. There are two major types of epitopes: continuous epitopes and discontinuous epitopes. A continuous epitope, also known as a linear epitope, is formed by a continuous sequence of amino acids. The antibody recognizes amino acids that are next to each other in primary protein sequence. However, many epitopes are discontinuous epitopes, also called conformational epitopes. In this case, amino acids that are separated in linear sequence come close together when protein folds into its three-dimensional structure. The antibody recognizes this specific three-dimensional shape. This explains why protein folding and structure are extremely important for antibody-antigen interaction. The part of the antibody that directly binds the epitope is called antigen-binding site or paratope. The antigen-binding site is located at the tip of each antibody arm. Because an antibody has two arms, most antibody molecules have two identical antigen-binding sites. The binding between an antibody and antigen is highly specific. This specificity depends on the shape, charge, and chemical properties of the antigen binding site, matching the epitope, similar to a lock and key interaction. Now let's look deeper into the antigen binding site. Within the variable regions of both heavy and light chains, there are small areas with extremely high variability. These areas are called hypervariable regions. If I draw the hypervariable region of the heavy chain as a single line, you can see three hypervariable regions which are highlighted in red in the diagram to your right. Similarly, the diagram below shows the hypervariable regions of the light chain. They are also known as complementarity-determining regions or CDRs. Each variable region contains three CDRs: CDR1, CDR2, and CDR3. So CDR and HV are the same thing. These regions show the greatest variation between different antibodies. When the antibody folds into its three-dimensional structure, these CDR regions come together and form the antigen binding surface. Therefore, the CDRs are the most important regions responsible for determining antibody specificity. Between these CDR regions are more conserved areas called framework regions or FRs. There are four framework regions: FR1, FR2, FR3, and FR4. The role of framework regions is to provide structural support and correctly position the CDRs so they can interact with the antigen. Next, let's discuss antibody fragments. There are different functional parts of antibodies by cutting antibodies with specific enzymes. When an antibody is treated with enzyme papain, it produces three fragments. Two of these fragments contain antigen binding sites and are called FAB fragments. FAB stands for fragment antigen binding. Each FAB fragment contains one complete antigen binding site. The remaining fragment does not bind antigen but contains the constant regions of the heavy chains. This region is called FC fragment, which stands for fragment crystallizable. The FC region is very important because it communicates with other components of the immune system. Another enzyme called pepsin cuts antibodies differently. Instead of separating two antigen binding arms, pepsin keeps them connected. This produces a larger fragment called FAB2. FAB2 contains two antigen binding sites linked together. Now let's focus on the Fc region and Fc receptors. Fc receptors or FCRs are the receptors found on the surface of many immune cells, including macrophages, neutrophils, and natural killer cells. In the diagram on your right, you can see Fc receptors shown in green expressed on macrophages. These receptors bind to the Fc portion of antibodies. When antibodies are freely circulating without binding to an antigen, they interact very weakly with Fc receptors. However, when antibodies bind to the antigens on the surface of a pathogen, many antibodies become clustered together. This allows multiple Fc regions to bind Fc receptors strongly. This interaction activates immune cells, in this case macrophage. For example, when Fc receptors on macrophages bind antibodies coating bacteria, the macrophages become activated and engulf the bacteria through phagocytosis. Therefore, the FAB region recognizes the antigenic epitope, while the Fc region communicates with the immune system to eliminate the pathogen. Now let's discuss the 5 major antibody isotypes. Humans have 5 major classes of antibodies: IgM, IgD, IgG, IgE, and IgA. The main difference between these antibody classes is found in the constant region of the heavy chain. Different heavy chain constant regions give antibodies different structures and functions. These different heavy chains are called isotypes and are designated by the Greek letters mu, delta, gamma, epsilon, and alpha. For example, an antibody with a mu heavy chain is called IgM, while the one with a delta heavy chain is called IgD, and so on. It is also important to know that some antibody classes have subclasses. IgG has 4 subclasses: IgG1, IgG2, IgG3, and IgG4, while IgA has 2 subclasses: IgA1 and IgA2. Although all antibodies are built from the same basic unit of 2 heavy chains and 2 light chains, IgM and IgA can join together to form larger molecules. IgM usually exists as a pentamer consisting of 5 antibody units, while IgA commonly exists as a dimer consisting of 2 antibody units. These antibody units are linked together by an additional protein called the J chain. Because IgM and IgA contain multiple antigen binding sites, they can bind to several identical epitopes on the same pathogen at the same time. This greatly increases their overall binding strength, known as avidity, making them particularly effective at neutralizing pathogens. Let's briefly look at the functions of each antibody class. IgM is usually the first antibody produced during a primary immune response. IgD is mainly found on the surface of naive B cells, where it functions as part of the B cell receptor. IgG is the most abundant antibody in blood and tissues, provides long-term protection, and is the only antibody class that can cross the placenta to provide passive immunity to the fetus. IgE is involved in allergic reactions and plays an important role in immunity against parasitic infections. Finally, IgA is a major antibody found at the mucosal surfaces, including the respiratory and gastrointestinal tracts, where it provides a first line of antibody defense against invading pathogens. So in summary, I explain you 4 topics in this video presentation. In terms of basic structure of antibodies, heavy and light chains, variable and constant regions, VL, VH, CL, CH, and antigen binding sites were explained. In terms of how antibodies recognize antigens, antigen versus epitopes, continuous versus discontinuous epitopes, paratope, CDRs, and framework regions were explained. In terms of functional regions of antibodies, I explain you FAB, FC, FAB2, and FC receptors. Finally, in terms of antibody isotopes, I explain you IgM, IgD, IgG, IgE, and IgA. I hope this video is helpful for you. If you like it, please subscribe and share. I greatly appreciate it. Thank you and have a nice day.